Hi, let's talk about single-cell RNA sequencing, short for scRNA-seq. Why we need to use single-cell RNA sequencing? For each tissue, there could be many different types of cells, and we want to know individual cell types and what's their genetic sequences. So to do this, we will use a technique. It's called a single-cell RNA sequencing. Using this technique, we'll be able to get gene expression, for example, gene 1, gene 2, until gene N, and the expression levels for individual cell, like cell 1, cell 2, cell 3, cell 4, until cell N. We'll get a very large matrix, use a barcode.tsv file, represent the cell names, and the gene names will be stored in the genes.tsv file, and the large matrix we mentioned will be stored as we call them counts. After you loaded the data, it will look like something as a big table. Even though it's a sparse matrix, but it will understand this one as a matrix. Next, we'll go to Seurat, directly load the files and see what the data format will look like. Once you download the data, you'll find 3 files, which are barcodes, genes, and matrix. You can open these files using text editor. In the barcode, which are just the cell names, you can go through and see each barcode represents one single individual cell. The gene.tsv file, it is stored 2 columns. The first column would be the gene ensemble name, which normally would be hard to recognize. So we normally use the second column as a gene name or gene ID. Third one is called matrix.mtx file. It is a matrix that kind of stored in a sparse way. The first 2 columns are coordinate. This is the coordinate of the gene, and this is the coordinate of the cell. The third column represents the gene count, which means the first cell and the 32,709 genes expression level is 4. It has 4 copies, and we'll load the 3 files using Seurat. Let's open RStudio. In our previous video, we have talked about how to install Seurat in RStudio. Here, let's use library Seurat to import the package. Click Run. Then let's load the data. The data loading will be very simple. We can first create a variable name called a Seurat.data. The dot doesn't mean anything. It's just for convenience to remember the name of a variable. To assign the value, we use a less than and a dash represent we want to assign something to the Seurat data. We can simply use read 10x. Only need to provide the directory of the folder. So here it's going to be d-data. Use a quotation mark. If you click run, you'll see there is unrecognized escape character string. So under Windows, we'll need to change the / to / and run it again. Once we load the data, we'll see the Seurat.data will be show up as a variable. If you click the arrow, so these will be the raw data that read directly using the read 10x. It will directly read the barcode, genes, and matrix. If you want to know specific function and its description in R, you can simply use a question mark followed by the read 10x. The description of the method will pop up on the right side, and you'll see this is a method that enables easy loading of sparse data matrix provided by 10x Genomics, right? So we only provide the data.directory, and the default value for the gene column is the second one, as we mentioned in the gene.tsv file. As we mentioned in the gene.tsv file, the second column is the gene name, which will be much easier to recognize, right? And the cell column would be the first one. The cell column owning in the cell, which is the barcodes.tsv file, only contains one single column, so that will be the cell name. The cell name will be the barcode. Let's create a Seurat object. The function we will use is called create Seurat object. An object we need to provide it, as we mentioned, it's a count. The count is a matrix. It will come from the raw data we just loaded. So we'll name it as a Seurat.data. We can name the project as any kind of project we prefer. Here we can name it as Seurat project. And you can hit the run. It will load the data. Because the gene names in the raw files contain some underscores, and as this warning shows up, the function will replace the underscore with the dash to avoid any kind of misinterpretation of the data. Now we can get the second variable in the environment, which is, we just name it as a Seurat object. If you click the drop-down menu, you'll see it has a lot of information, which is kind of overwhelming. They are normally called slots. Slots can be, think about, it's a different location in the same kind of object, and then we can store different type of data in different slots. So the slots available in the Seurat object are listed here, which are assays, metadata, active assay, active identity. But how can we get access to these slots? We can basically use $sign, for example, Seurat object, and the $sign, and called assay, which is the first slot, and we can hit run. This one will shows the message called it's RNA. The assay is RNA, and the data contains 32,000 features for 2,700 cells. The first 10 features, which the features are equivalent to genes here. So these are first 10 genes, and layers we use the counts, basically just the number of gene copies for individual cells. We can also extract information from other, for example, metadata. The metadata will describe the different cells. These are cell names, and this is the number of counts. So if we use Seurat object at metadata, we'll get information for each individual cells regarding their number of counts of RNAs and the number of feature RNAs. The difference between these two would be the second column represent a smaller number. They are unique genes. So all the genes, which means in this cell, there are 781 different kinds of genes, but each gene can have multiple copies. So the total copy number will be 2,421. So that's the difference between these two. And this is how we can get access to this information. So when we do other analysis, the information will be stored in this Seurat object as a whole, and you can extract information from this object. So in our next video, we'll talk about the first pre-processing of the Seurat object.